Our next speaker will be Sydney Wizenberg, presenting Monitoring Phenological Changes via the Plant-Pollinator Interface.
Thank you for having me, and thank you for the warm welcome, Laura. Lovely, thank you for having me. So a small introduction, my name is Sydney. I'm a postdoctoral fellow at Naturalis Biodiversity Center in the Netherlands. But before I started my position here, I was a postdoc in Amro Zayed's lab at York University. So I'm a big fan of Beacon, and I think I'll be here for many years to come. So today I'm going to be talking about a project that I worked on last summer when I was at York. It's called Monitoring Phenological Changes via the Plant-Pollinator Interface. So what is phenology, first of all? This is probably a familiar term for some people in the room, but it may not be a familiar term for everybody in the room. And phenology is kind of a broad term that refers to general cyclical nature of natural phenomenon. So a great example of this is leaf senescence in the fall, but we can also think of phenology as transitioning through the life cycle, typically of plants and animals. So when we're talking about plants, and I know this is a room full of entomologists, so you guys are probably thinking of bug life cycles when you talk about this, but I am a plant biologist, so plants are the first things that come to mind to me. And when we talk about the plant life cycle and phenological timing, we're talking about the temporal transition through these different stages of the plant life cycle. So that'll be germination from a seed into a small vegetative plant and eventually a flowering plant that then fruits and sets seed. So this transition through the life cycle actually occurs in response to environmental conditions. So phenological timing and transition between stages of the life cycle is environmentally dependent. It's kind of like a plastic phenotypic trait. And what that means is it varies in response to the environment as well. So synchronized phenology and plant-pollinator interactions are very, very important. And that's because plant-pollinator interactions rely on synchronized phenology. So we have to have an overlap in the timing of their life cycles for a plant-pollinator interaction to take place. And that's because this synchronized development is really important because non-adult pollinators typically won't forage on pollen, and plants only produce pollen when they're flowering. So you can kind of think of the life cycles as happening concurrently, and there's this small temporal overlap, and that is this really important precursor for a successful plant-pollinator interaction. So because phenological timing is a plastic phenotypic trait, environmental changes will produce phenological changes. We already know this to be true, and this can occur in response to things like rising temperatures, erratic weather patterns, landscape conversion, and urbanization. And that's because all of these changes disrupt the abiotic cues that trigger phenological processes in plants and pollinators. So a great example of this is that I was talking about leaf senescence previously, which is that change of colors that we see in the fall. And that occurs in response to the drop in temperature that we typically see in autumn as well. So if fall arrives later, we will also see delayed leaf senescence. Same with if spring arrives early, we may see earlier flowering in plants and also earlier germination of seeds in the ground. So phenological changes could disrupt pollination services. So you should understand by now that this temporal overlap is really important for plant-pollinator interactions and that environmental change is going to produce phenological change. And what that can look like is disrupted phenological timing. So in the case of plant-pollinator interaction, sorry, I'm wondering if I can move that little bar. I don't think I can. Hopefully everyone can see this here. But if insect phenology advances and plant phenology does not, we could get temporal mismatch. So you could see here that if insects start developing earlier in the spring, but plants do not, and by the time that they're adults, the plant may not be flowering yet, and thus there's temporal mismatch, and this prevents pollination. The same can occur in the reverse, which is that plant phenology can advance and insect phenology may not. And this is, again, an example of desynchronization that prevents pollination. So the overlap in timing in development for plants and pollinators is really important. And if we see any shift in either of these groups independently of the other, this can disrupt pollination services. So the direction and pace of phenological shifts are really hard to predict. Now, if you're familiar with this particular subfield of biology, you may know that generally speaking, we see plants flowering earlier as global warming is occurring. But we can't really make generalization across species or even across communities because we know that this can vary across fine and large spatial scales. And a really great example of this, it was a 33-year study done in Arizona that was published in Current Biology back in 2020. This is an amazing paper. I really recommend reading it if you're interested in this topic. But what the authors did is they actually hiked a trail along a mountain in Arizona for 33 years and completed plant surveys, taking note of the first date of flowering of a collection of plant species that they were studying. Now, when they grouped all 5 of these sites together, so this was actually an altitude gradient along the mountain, they saw that there was no real phenological shifts. But when they broke down the habitat patches based off of altitude, what they found is that the change in flowering time varied both in the direction and pace of change across habitat patches. So at the lowest habitat patch, which is site 1 down here, so the very bottom of the mountain, it did get warmer across 33 years, and flowering actually advanced. So the plant started flowering on average 20 days earlier when we look at the differences between the first and last year of the study. We moved up to site 2, they were still flowering earlier, but at a lower degree. And then we saw a shift at sites 3 and 4, where flowering was actually delayed. And then finally, site 5, which was at the very, very top of the mountain, this was a non-significant change in flowering time. So if you group all of these and pool all the sites together, it doesn't look like there's any phenological change happening. And really what this shows us is that even fine spatial scales can elicit different phenological shifts. And that really is the crux of the issue here, is that these are very hard to predict. We can't make generalizations, and we don't really know how this is going to impact plant-pollinator relationships because it's so hard to predict in the first place. So large-scale monitoring is really the only approach to decode this complex phenomenon. We know that it's complicated, and we know that it's going to vary a lot, and studying these relationships are really the only way to understand how this could take place and what the impacts could be. But traditional ground-based techniques are laborious and really limited in their scope. You can kind of see that in this paper here, that they had to physically survey this area for 33 years. They weren't able to cover a large amount of ground. And because of that, there was a lot of time invested into this project, and it makes it difficult to try and track these trends on a really large scale. So we kind of asked this question last year. It was when I first started working at York almost 2 years ago now, that I first started working on pollen metabarcoding for a project called BCSI. And as a plant biologist coming into an entomology lab, the first thing I thought of is, oh, the bees are telling us when plants are flowering. And Amro thought that was a little bit crazy, but he told me I could design a pilot project, test it out with a couple of research assistants. He'd pay for 2 sequencing runs, and we could see if honeybees could actually act as research assistants to track plant flowering phenology. So we asked if metabarcoding could really be the solution to modernizing this field, knowing that we needed to come up with a new approach that would be high throughput and allow us to monitor this on a large scale. So we specifically set out to test if multi-locus metabarcoding of pollen collected by bees could be used to infer the flowering phenology of plant communities by a comparison to in situ field monitoring. So what that meant is that I was physically in the field around the Lumbers building at York doing plant surveys last summer for 4 weeks. We were simultaneously operating pollen traps at our rooftop apiary, collecting pollen to understand if the plants we saw flowering in the field were also showing up in our pollen samples, and if there was any potential time delay between when a plant started flowering and when we would expect it to show up in our pollen samples. So what we found is that, first of all, there was a lot of genera that were flowering at our monitoring site that weren't present in our pollen metabarcoding libraries. That's not very surprising. There was also lots of genera detected in our pollen metabarcoding libraries that I did not survey on the site. That was also not surprising because we only surveyed a 1 kilometer radius around the rooftop apiary, which is really just a physical limitation of the project. And honeybees will generally forage outside of that range. So that was kind of expected going into it. But for the 34 genera that occurred in both data sets, we found that the honeybees could reliably indicate the onset of flowering, but not the length nor the duration. So in this graph on the left here, we've actually overlaid flowering type or time as measured in the field. So that's the colored bar. And then the black bar is when we detected pollen in our pollen samples for the 4 weeks that we were monitoring both via the honeybees and in the field. So you can see here that there was actually an 100% rate agreement that when something started flowering in the field, if the honeybees were foraging on it, they were really good at picking up on the pollen right away. And we saw it in our pollen metabarcoding libraries. Where they lost a little bit of their reliability was their ability to indicate the length or duration of flowering. So they would quickly lose interest in a lot of species like Tilia, which was the little leaf lindens on York's campus. They only foraged on those for the first week that they were flowering. Then they quickly lost interest and moved on to something else. Now, interestingly enough, there was a kind of a subgroup of species, particularly within Rosaceae, so flowering cherry and apple trees, that honeybees tended to be good at indicating the entire length of the flowering period, but this wasn't consistent enough to be able to track this on a large scale. So they can indicate the start of flowering, but not the end nor the duration. We also saw that genera detection rates increased with the number of loci and colonies used for monitoring, but they did not display a notable asymptote, and that was kind of interesting as well. So unsurprisingly, if you use 5 colonies instead of 1, you detect more plant species, and if you use 2 loci instead of 1, you detect more plant species. What we would expect here with species detection rates is for this to eventually level off, and we didn't pick up on that in this project with 2 loci and 5 colonies. So that suggests that you could potentially deploy more honeybee colonies and more loci for monitoring and yield even greater species detection rates. Now, of the 5 colonies we used for monitoring, 2 of them ceased collecting usable amounts of pollen towards the end of the summer. They were 2 smaller colonies. But for the 5 colonies that had complete time series for all 15 weeks that we were operating the pollen traps, we found that there was really similar diets at upper taxonomic levels. This is good news because it suggests that the colony level effect is not that strong here. So colony turnover won't necessarily discredit the integrity of a monitoring program. If the end game here is to set up a permanent monitoring site, we know that honeybee colonies do collapse and will need to be replaced. And one serious consideration here is how that would impact our findings. But this suggests that the colony level effect is not strong enough to seriously disrupt our ability to use this tool. So we successfully inferred the onset of flowering for 222 genera in this 15-week monitoring program. So that was a lot of plant genera that we picked up on here. And I do want to note that I don't typically barcode at the species level. So we look at 2 loci signal detections at the genus level. And this suggests that even though they don't forage on every plant species that was present at the site, they certainly can survey a lot of plant genera and a lot of plant families, meaning that this kind of super generalist behavior of honeybees makes them uniquely qualified for this type of work. We also looked at flowering genera density at upper taxonomic levels. So these ridge plots over here indicate how many genera within that taxonomic order started flowering during that week. And what we can see here is there's some preliminary evidence of phylogenetic clustering of flowering behavior. Now, because this is only a single monitoring program, so a single year at a single site, we can't draw that conclusion more broadly, but it suggests that we may see this trend in other future monitoring programs, and that there could be some phylogenetic basis to flowering time within plant families and orders. So in conclusion, honeybees are fairly reliable research assistants. They're really good at indicating the start of flowering, but only in species they selectively forage on. That being said, what we saw here was they forage on a lot of plant species. So they're not going to forage on everything at a site, but they're going to forage on more than you would probably expect. And we can therefore track phenological trends for those plant species, with the one caveat here being that you can't draw any conclusions about species not present in your metabarcoding libraries. So if a plant is available at the site and the honeybees don't forage on it, we can't say that it wasn't flowering, for example. We just can't track its flowering time via honeybees. We also saw that honeybees are uniquely qualified for this work. So the colony level effect was not very strong, which is really good news, taking into consideration colony turnover between years of monitoring. And also that they really are super generalists. I don't throw that word around loosely, but we've done a lot of work using pollen metabarcoding on pollen collected by bees and consistently see that they're foraging on a lot of plant species all the time. So they're really good at surveying communities of flowering plants. We also saw that the start of flowering may be associated with plant phylogeny. This is some early evidence of this, but we can't draw any major conclusions yet based off of this single project. I also wanted to note here that sharing of these results is welcome. That's why I didn't put the no photos up on the slide, but please do cite our preprint that's up on bioRxiv, and you can find the link to this on my Google Scholar page. So finally, I'd like to give some special thanks to the Zayed Lab at York University. This work was supported by an NSERC Discovery Grant awarded to AMRO and a York Genomics Research Chair held by AMRO. I'd also like to thank the Evolutionary Ecology Group at Naturalis, which is where I work currently. This work was also supported by the Biodiversity Plus Project Meta Plant Code, awarded to Barbara Gravendale. They are currently paying my salary while I work on this project. So I'd like to thank them as well for their contributions to this work.